Good morning, student. Today we discuss about the serology and immunology. In serology and immunology, specifically we discuss about the techniques. Okay, because introduction of the serology and immunology we already discussed in the previous video. So in this video we discuss about the technique. So first of all, what is immunoprecipitation reaction? Actually, in immunology, all reaction will takes place only between antigen and antibody, nothing else. All reaction will take place only in antigen and antibody. So don't confuse about that. If any question will be asked, what is immunological reaction, what is immunoprecipitation reaction, what is agglutination reaction, and in which substance this reaction will occur, don't confuse. Only that answer is the antigen and antibody. All reaction will take place between antigen and antibody. Okay, so immunoprecipitation reaction is actually is a highly specific interaction that occur between the antigen and antibody. You already know about the enzyme and substrate reaction. How enzyme will react with substrate, and how they initiate or activate the reaction of normal reaction. How the enzyme activate the normal reaction, and how they decrease the activation energy or time. So similarly, it is like enzyme and substrate association. Okay, because in enzyme, you know that enzyme having a site where substrate come and bind. After binding the substrate with the enzyme, they convert it into a product and enzyme will remove. Okay. Similarly, antibody, in previous slide we discussed about the structure of the antibody, that is a Y-shaped structure, right? In Y-shaped structure, it consists of two heavy chain and two light chain. Okay, this is a light chain and this is a heavy chain, and this region is called Fab region. What region? This is a Fab region where antigen come and bind. Okay, so similar here, enzyme having a site where substrate come and bind. Here antibody have a site. Antibody having a site where antigen came and bind with it. Okay, and the site where antigen came and bind with the antibody, that site are called paratope. Paratope in antibody, and epitope in antigen, because antigen also having a site where they go and bind with the antibody. Okay, so every paratope is very specific to epitope. Okay, and they interact with each other with a non-covalent interaction or van der Waals interaction, hydrogen bond, van der Waals bond. Okay, so the binding between antigen and antibody involve weak and reversible non-covalent interaction, right? Weak and reversible non-covalent interaction. Reversible means what? Reversible means when antibody combine with antigen, they form a antigen antibody complex, right? They form antigen antibody complex. After that they again separate and convert into antigen plus antibody. Okay, This process is called reversible process. Okay, this process is called reversible process. Means they form a complex, again they separate and converted into a enzyme or substrate or in a substrate reaction. Okay, this is a weak and reversible non-covalent interaction. What type of interaction will take place? What type of interaction? Means this paratope and epitope will bind with each other. What type of interaction will take place? That is a non-covalent interaction. Okay, in non-covalent interaction, what type of that non-covalent interaction? This is a van der Waals forces or hydrogen bonding or hydrophobic forces. Okay, what type of forces? Van der Waals force, hydrogen bonding or hydrophobic forces. Next, the smallest unit of antigen that is capable of binding with antibody is called an antigen determinant. Already I discussed antigen determinant. What is an antigen determinant? Nothing else. It is a epitope. Okay, it is a epitope. See, this is an antigen, this is an antibody. The site they came and bind with this site is called this site is called epitope. This site is called epitope. Okay, and this site is called paratope, and this epitope is also known as antigen determinant. In question, maybe MCQ, they ask that what is the antigen determinant. Don't confuse. It is nothing else. It is a epitope. They give option are there. First is paratope. Second is Fab region. Third is epitope. Fourth is none of the above. Nothing else. It is a epitope. Okay, it's clear. See here diagram. This is antigen. Okay, this is antibody. Okay, this is antibody. Here is a site in area, in blue area, that is a paratope, and here that region which come out with this circle, this is a epitope. Okay, they combine with each other and form a antigen antibody complex. Okay, clear? This slide is clear? So immunology, what is immunological reaction? Immunological reaction is an antigen antibody interaction that takes place by a non-covalent interaction, weak non-covalent interaction. Okay, in weak non-covalent interaction includes like van der Waals forces, hydrogen bonding, hydrophobic bonding, and the area where they go and bind with each other are epitope and paratope. Okay. So next slide is a immunoprecipitation reaction. I already told that in immunology, all reaction will takes place only between antigen and antibody. It is immunoprecipitation reaction, or it is immunoagglutination reaction, or other reaction. Every reaction will take place between only antigen and antibody. Okay. See here, precipitation occur maximally only when there are optimal proportion. This is a very important. What? Optimal proportion of the two reacting substance, antigen and antibody. Means what? Precipitation precipitation reaction will only takes place when a fixed proportion of antigen and antibody are present in a solution. Only when fixed means antigen into ratio of antigen to antibody is fixed or optimum. Only this condition precipitation will take place. Okay. If antigen is excess, then it is known as zone of antigen excess, or if antibody is excess, means it is a zone of antibody excess. Okay. And where the optimum ratio is present and precipitation will take place, that zone is known as zone of equivalence. Zone of equivalence. Okay, zone of equivalence, where ratio of antigen and antibody is fixed or optimum. That zone is known as zone of equivalence. Hence, an absolute antibody antigen complex formation occur within a narrow range known as zone of equivalence. And the curve by which this this type of zone we determine this type of zone, that curve is known as precipitin curve. That curve is known as precipitin curve. See here. Here antigen and antibody both are present in this area. Antigen and antibody both are present, right? But in this area, antigen and antibody both are present, but in this area, see here, but in this area, ratio of antibody is more as compared to antigen. So, this area is known as zone of antibody axis. Here, the ratio of antibody and antigen, actually they are equal, because antibody having two paratope, okay, one antibody having two paratope. So we can't say that number of antibody equal to number of antigen. This is a zone of equivalence. This is known as zone of equivalence. No, this is wrong. We can't say that zone of equivalence depend on the number of antigen and antibody. No, it depend on the optimal proportion, because one antibody having two paratope, some antigen having more than one epitope. So it is the optimal proportion of the two reacting substance, antigen and antibody. That optimal proportion where they occur in a reaction, that step or zone is known as zone of equivalence, where neither antigen is in a excess amount or nor antigen in a excess amount. Okay, that area is known as zone of equivalence. Another is a zone of antigen axis. See here. Here is antigen in a excess amount. Here antigen axis, here antigen axis, antigen axis. So this is a zone of antigen axis. And the curve, this is a curve by which determine the zone of equivalence. This curve is known as a this curve is known as a precipitin curve. Okay, this curve is known as precipitin curve. Actually, precipitin curve is plotted between the antigen and antibody. Okay, in x-axis antigen is added, and in y-axis antibody precipitated. Okay, clear? Don't confuse with the that zone of equivalence is ratio where number of antigen is equal to antibody. No, we can't say that number of antigen is equal to antibody, or a number of antibody is equal to antigen. Okay. Another first technique, that is a radial immunodiffusion. What is a radial immunodiffusion? Radial immunodiffusion, it is a technique by which we have to measure the concentration of an antigen. What we measure? We measure the concentration of an antigen. Okay, concentration of the antigen. How we can determine the concentration of the antigen? By we determine the concentration of antigen by measuring the area. By measuring the area. Okay. The relative concentration of an antigen can be determined by a simple quantitative assay in which an antigen sample is placed in a well and allowed to diffuse into a agar gel containing antibody. Okay. The diameter of the ring is proportional to the log of the concentration of the antigen, since the amount of antibody is constant. See here. This is the agar plate. Okay. This is the agar plate. Because why we take agar plate? Because in solid media we can easily determine the concentration of antigen. Okay. This is the agar plate. Agar plate containing blood antibody. When we make a solution of agar, in that solution we mix the antibody. Okay. Suppose number one is a person. They having a flu virus. Okay, they having a flu virus. We have to determine or detect that virus, if they are present in that person or not. How can we determine? For this flu virus, we have to antibody, right? We have antibody. So this antibody are mixed in a agar plate. Because we have to determine the specific antigen, concentration of a specific antigen. So now this is a agar plate which contain antibody. Okay. Now we punch the gel. Okay. After punching, a well is created. In this well, we add the specimen of sample of this person. Okay. Sample. We add the sample. Okay. If this person having the antigen against this antibody, then they diffuse or react with this antibody and form a complex. They react and form a antigen-antibody complex, right? And after forming the antigen-antibody complex, a precipitin ring is formed. This is a precipitin ring. This is a precipitin ring where precipitation will take place, right? This is a precipitin ring, and this is a zone of equivalence. Zone of equivalence. This is a zone of equivalence where optimal proportion of the antigen and antibody complex will take place. There is nothing will access here. Nothing will access neither antibody nor antigen. Okay. In this area, antibody is axis, right? And in this area antigen is in axis amount, right? Here antibody is in axis amount and here antigen is in axis amount, and this is a ring where complex will be formed and zone of equivalence. After determining the area of the zone of equivalence, we know the concentration of the antigen and determine the concentration of the antigen. How much that person is infected and how much they need the antibiotic or else how they treatment will takes place. So this is a radial immunodiffusion. This technique is a radial immunodiffusion. Okay. Another technique is a octolony or a double diffusion method. Actually, in this method, only the movement of antigen will take place and antibody is in a constant manner. Where in this method, both antigen and antibody will move. Okay. Both antigen and antibody will move. See, this is the octolony double diffusion method. What is octolony double diffusion method? Double diffusion means both antigen and antibody will move in a gel. Okay. Both antigen and antibody will move in a gel. See, both antigen and antibody diffuse radially from wells toward each other. Means what diffuse radially toward each other means both the movement of both antigen and antibody will take place and they form a complex, antigen antibody complex, thereby stabilizing a concentration gradient. They form a concentration gradient. Antigen and antibody diffuse toward each other at the rate that increases in proportion to their concentration in the well, but decrease in a proportion to their size. It is a immunological technique used for the detection, identification and quantification of antibody and antigen. By this method, we easily identify the antigen and antibody both. In radial immunodiffusion, we use only one type of antigen, but in double immunodiffusion, we can use more than one type of antigen. See here, this is a agar plate. This agar plate only contain agar, not antibody. Remember that. In radial immunodiffusion, agar plate contain antibody, but in double immunodiffusion, agar plate not contain antibody. Okay. This is a agar plate. We punch the agar plate and form a well. See, this is center well and this is a side well. This center well having a antibody. We put antibody in a center well, okay, and inside well we put the antigen, different types of antigen. Now we have to determine this antibody having paratope for this all type of antigen or a single type of antigen, okay, or the antigen having epitope for this antibody or not, we determine. When after few minutes we see that some type of identity will form, like cross like identity or partial curve like identity, or cross and partial curve both will take place. This type of identity will be formed, okay, when antigen antibody complex will takes place. When antigen antibody complex will be formed, okay. So what type of what determine, how can we determine this identity, and what they specify, like what non-identity, which type of reaction they specify non-identity. So non-identity means, or non-identity occur when two antigens are unrelated. Means these two antigen, this antigen 1 and antigen 2, these two antigen are non-related or not related to each other, not related to each other, not related to each other, then this non-identity will be formed. If these two antigen are not related to each other, totally they are totally different to each other, then non-identity will be formed. Okay, means they share no common epitope. Means if this antigen bind to this antibody, then this antigen will not bind to this antibody, because there is no common epitope in these two antigen. Antigen 1 and antigen 2 having no common epitope or similarity, so they having non-identity. What do you mean by identity? This is identity. What do you mean by identity? Identity means two antigens share identical epitope. Two antigen share identical common epitope means this antibody bind with both antigen 1 and antigen 2. See, if this antibody bind with this antigen 1 and this antigen 2, if antigen 1 and 2 both are bind with this antibody, means means both antigen having common epitope for this antibody, right? This means they are identical to each other, or they form a identity, or they form a like a curve. This come under identity. And third one is a partial identity. Partial identity occur when two antigen share common epitope, but one of the other has a unique epitope. Means both antigen, means antigen 1 and antigen 2 both share common epitope with this antibody, right? Both share common epitope, but this antigen 2 having some another type of epitope, they having also some another type of epitope. Okay, understand? Means see here, antigen 1, antigen 2 means both having same epitope for antibody, this star mark, for this star mark antibody, both have a common epitope for this star mark antibody, but this antibody 2 having some another epitope, means epitope this star mark epitope for this star mark antibody, and they having this epitope also, this type of identity is known as partial identity. Means partial identity occur when two antigens share common epitope, but one of the other has a unique epitope. Okay. The anti-serum form a line of identity with a common epitope and a curve is spurred with a unique epitope. Okay. Means curve is spurred with a common epitope. Like this type of curve is spurred is for the common epitope, like this identity. Okay. And form a line for the unique epitope, like this line and this curve. Okay. For this two type of cilia. This is for common epitope and this is for unique epitope, right? This is for unique epitope. So don't confuse with line and curve. Curve is for common epitope and line is for unique epitope in a partial identity. Okay, clear? Another is a agglutination reaction. What is a agglutination reaction? See what is a agglutination reaction? Actually, agglutination will take place when a visible clump will be formed. Agglutination reaction will take place when visible clump will be formed. See, when antibody complex will be formed, this complex is in a visible form. Visible clump is in a visible clump. This is a agglutination reaction. Agglutination reaction is the visible clumping of a particular antigen. Is a visible clumping of a particular antigen when mixed with antibody specific for the particular antigen. Okay. Antibody that produce such reaction are called agglutinin. Okay. This antibody which produce this type of visible clump is known as agglutinin. This is known as agglutinin. Actually, there are five isotype of antibody. Antibody having 5 isotype. In previous video, we already discussed about the 5 isotype, right? Like IgE, IgD, IgA, IgM and IgG. In this isotype, IgM having 10 valency, right? IgM having 10 valency, or this is the highest valency of all isotype. IgM having the highest number of valency. So IgM having highest number of valency means they are a best or they are a good agglutinin. More valency means more antigen came and bind. So more antigen come and bind with the antibody means the agglutination reaction or visible clump will be formed more clearly. So this reaction is a very good reaction with IgM. Okay. There are two type of agglutination reaction. First is direct agglutination reaction, another is indirect agglutination reaction. Actually, indirect agglutination reaction not and serum will be not we can't isolate in this indirect agglutination reaction. We take only blood sample with erythrocytes. Erythrocytes already having an antibody and we add that in that antibody we add anti-IgG reagent and that reagent will agglutinate this erythrocyte sample and we see the clumping. This type of method is known as direct agglutination reaction. And indirect agglutination reaction, we take a serum. that contain antibody, serum having antibody, and that serum having antibody fused with the erythrocyte cells that are only erythrocytes. No, they don't have a antibody. After fusing with erythrocytes, anti-IgG will be added in this sample, and agglutination reaction will take place, and clumping will form. Right? This type of reaction is known as indirect agglutination reaction. So another assay is immunoassay. Immunoassay is based on a antigen specific reaction between the antigen and antibody. Immunoassay is based on the specific reaction between the antigen and antibody. Immunoassay can be competitive type and non-competitive type. Actually, immunoassay is of two types: competitive type immunoassay and non-competitive type immunoassay. In competitive type immunoassay, see here, in competitive type immunoassay, antibody are attached with this sample, okay, and labeled and unlabeled antigen, they fused with the labeled and unlabeled antigen. See, this is unlabeled antigen and this is a labeled antigen. This is a label antigen, okay, fused with this type of antigen. After that, after this fusion, we easily assay this label antigen. Easily assay this because fluorescent color will be produced by this antigen, and we easily determine this antigen. This type of assay is known as competitive immunoassay. In non-competitive immunoassay, we again add the, see here, this is an antibody, and we add unlabeled antigen in non-competitive type, unlabeled antigen, and we add another antibody like another antibody that having labeled antigen, okay, that having labeled antigen. They come and bind and produce fluorescent light, okay. In this is a non-competitive, this is a competitive, because we add label and unlabeled both. The competition will take place in this antigen which go and bind with this antibody, okay. Here is no competition which takes place, all are unlabeled antigen, okay. See here, in non-competitive immunoassay, directly measure antigen. In non-competitive, we directly measure antigen. Means we directly add antibody with label antigen and measure it occupied site. While in competitive assay are based on the detection of total and unoccupied site by the antigen. In competitive immunoassay, label and unlabeled antigen are simultaneously exposed to the antibody. Label and unlabeled antigen are simultaneously exposed to the antibody, right? And this type of immunoassay are used in ELISA. In next slide we discussed about in ELISA or in monoclonal in hybridoma technology. These both are based on the immunoassay. Okay, today we discuss till immunoassay only. Okay, in next slide in next video we discussed about the ELISA and hybridoma technology. Okay, thank you.